linking behavior with ecological and evolutionary processes.
Thank you, Mateo. Thank you very much for staying. Also, thank you for the invitation. It's been great so far. The work that I want to present to all of you today is part of the research we've been doing for last years of my postdoc. And basically what I try to emphasize is the importance of the underlying interactions when we study pattern formation, basically. And the way I'm going to be thinking about self-organization is as the process by which individual level interactions lead to this emergence of larger scale patterns. And if we have this definition in mind, then we may find different systems showing this self-organization in nature. This is true for systems with different properties and for these patterns covering different scales. For instance, we may have these collective behavior, collective movement patterns, both in microbial systems and also at larger scales. But also we may have this synchronization of behaviors affecting different traits rather than movement. For instance, in reproduction, and here we have the growth of colonies, both, again, in microbes and in macroorganisms. But even if we scale up to larger systems, we have these patterns that may cover up to hundreds of meters and sometimes even kilometers in this landscape patterning phenomenon. So as you can see, these systems are very different from each other, and then, therefore we should expect the mechanisms that are driving this formation of patterns to be different from one to the other. But there is something that is putting all these systems together, and it is that the emergence of these patterns is also going to lead to the emergence of new behaviors in the system. And just to give you a first example, that was actually the first one in which I started working and that has been motivating my work since, I want you to look at these vegetation patterns that we may actually find in very different regions all over the world and in ecosystems with very different properties. But despite all these possible places where we can actually find these patterns, their shapes show a kind of, they can be classified into actually very, very few classes. They usually fall in this class of spots, in which basically we have spots of vegetation that are somehow regularly distributed in the soil. But we may also have this more labyrinth or stripe of stripes. And finally, we also can find this kind of almost homogeneous layer of vegetation that has only some gaps of bare soil interspersed on it. So, of course, as people started as we started developing the ability of observe the Earth from above, and we started finding more and more of these patterns, people started wondering which could be the mechanisms that are actually forming these patterns. And they started proposing different ways of obtaining them, but one that actually was pretty successful and was very well accepted by the community is what is called the scale-dependent feedback mechanism. So very briefly explained with a picture, the scale-dependent feedback mechanism is telling us that the existence of vegetation has different effects on the growth of more vegetation depending on how far we are from that focal plant. So basically, when we have some vegetation and we are close to it, the presence of that vegetation is going to increase water infiltration rates, for instance. It's also going to decrease soil erosion processes, and therefore is going to locally increase the quality of the environment for more vegetation to grow. But as we move away from that plant, then we have a switch in the dominant interaction, and then it is the competition for resources what is actually dominating this interaction. And therefore, the presence of vegetation on larger scales has a negative effect from the growth of more vegetation. So how can we put this into math? Well, as maybe many of you are already thinking about, this mechanism is very similar to the activation-inhibition principle that was introduced by Turing last century. And therefore, the way that people started plugging this mechanism into equations is by creating a system of reaction diffusion equations in which we'll have an equation for the vegetation density, for amount of biomass that we have in the system, that will basically have a growth term here that is going to depend on water availability that is given by this W. We also may have some vegetation death, and we also have some kind of diffusion that is going to be given by how seeds disperse and how the vegetation spreads in space. But these dynamics, as you can see from this first growth term, is coupled to the availability of water. So we need a second equation for the amount of water that we have in the soil. And for this second equation, as you can see, we have second, we have this term here that is basically identical to this first one, that is actually the coupling between both densities. We also have some evaporation, so some loss of water, and we have some actual diffusion of water on the soil. And this first term here, it's actually going to account for the possibility of water coming from the above, from the surface to the soil, and therefore it's going to couple this soil water with the surface water density. So again, the surface water density is coupled to the soil water through this infiltration term, that in this case is identical. We also have some input that is basically rainfall, is a constant term that is going to account for amount of water that is externally introduced in the system. And we also have some water diffusion for this water. So once we have this closed set of equations, the way that the interactions are introduced is basically through this growth term. We have competition because the more vegetation we have here, the more water they are going to intake, and therefore the growth is going to be limited by the amount of water. But we also have some facilitation that is going to be given by this function here. So as you can see, if this, so basically this is an increasing function of the amount of vegetation, of P. So the more vegetation I have, the more infiltration I have, and therefore more resources I have to grow. Well, this is provided that W naught is smaller than 1, which is something that has been measured and is true. So in principle, these models have this interplay between competition and facilitation, and the way that these two interactions are acting at different scales is by the different diffusivities that different species have. So W is the amount of water, is the humidity in the soil, is how much water there is in the soil for these plants to be able to intake it via the roots and grow. It's all these are densities. Well, in the case of water, it's concentration of water here in the soil. This is. So
why is it linear? And why do you have such a linear W and you have other terms which have denominators with W? And where did these?
You mean these ones? Okay, so this is.
Are you assuming W is small or something like that?
So this first term is. It's basically if I plot this. I call this function, let's say, f of W. And this is W. This is a saturating function of W that basically has some half saturation constant that is K1. And is going, this constant is going to tell us how steep the growth curve it is as water increases, basically.
So you just want a function which has that kind of form.
Yeah, and actually, this is a function that is typically used, for instance, to model microbial growth on some substrate, and people are basically just plugging it for this model. And for the second term, it's pretty much the same. We have when P is equal to 0, then we have some baseline infiltration, and then as P increases, this goes basically to 1. So as I was saying, provided that we have these connections between diffusivities, then this model is able to recapitulate these patterns. But it's not only that, it also is going to reveal some interesting connections between the shapes of these patterns and processes that are happening at the scale of the ecosystem. So how can we look into that? The way of looking into that is basically by looking into how these models predict the mean vegetation biomass as a function of the availability of water in the system. So by looking into a phase diagram of this model. And when we look into these two variables, and here I mean what I'm going to take as an spatial average, we, as maybe not surprisingly, we observe a positive correlation between the amount of water and the amount of vegetation. So basically, more humid ecosystems are going to have more vegetation. But now if we relax this spatial average and we look into the actual shape of the patterns as we move across this gradient of rainfall, we observe that the shapes of these patterns is going to change consistently from having an homogeneous distribution to then the gaps, then the labyrinths, and finally the spots as the amount of water decreases. But even more interestingly, when we have I mean, when the system is showing this spot pattern, if the amount of water continues to decrease, then these models predict a catastrophic transition, and by catastrophic I mean that it's an abrupt transition into a desert state. So basically, once we are in this critical point, if the amount of water decreases, then the system collapses into a desert state. And by looking into this phase diagram, what this phase transition is also telling us is that once the system reaches the desert state, it's going to be very hard to recover it, because basically the amount of water, the rainfall is going to, I mean, it's going to need to increase up to this point to then be able to recover the original state.
So your equations are only valid until W equals 0, and then your equations break down. Is that what you're saying?
No, what I'm saying is when I look into the previous equations, and I basically, I obtain the phase diagram of the model, I obtain this bifurcation diagram, basically. I have an stable fixed point as the rainfall decreases. So basically, the control parameter here is the external rainfall.
So you say W never becomes negative if you just let those equations evolve?
No. At least for a set of parameters that have been kind of measured in the field, and then some sensitivity analysis, it doesn't. I don't know if for some combination probably it does, but not for. So as I was saying, there is something very important about these patterns, because basically this spot pattern is anticipating us something that is going to have very important effects for the ecosystem. So in principle, these kind of models are providing us with a mechanistic explanation for these patterns, but are also revealing some importance at the ecosystem level. So this is, yeah.
So the third is the solution of the new field theory without the diffusion.
Well, this is obtained numerically and averaging over space, but I think if you calculate the fixed points, it's going to be the same, because you have the effect of basically where is it? This facilitation term at the end. What is going to introduce you is this hysteresis loop here. It introduces an unstable fixed point.
Okay. And these are Turing patterns?
This is, sorry?
Turing patterns?
Yes. But as I was saying, in order to, because these are Turing-like patterns, it's very important to have that relationship between diffusivities. We need one species to diffuse much faster than the other. And this is something that hydrologists don't like a lot. The values that you need for the diffusivities to be in these patterns are much larger than the ones that they measure in the field. And also, although these interactions is true that they exist in nature, no one has been able to show in the field that they are the actual responsible of the patterns. So they are acting in this way, but there is no empirical evidence of them being responsible for the patterns. So then there is an important question that raises from these results, and it's whether this is the only mechanism that that can lead to these patterns, or whether we can find an alternative route to these structures. And as I started thinking about this problem, we actually started exploring the possibility of having these patterns only with one of these interactions. So basically, whether we can come up with a minimalistic model to obtain these results. So basically, what I'm going to show you now is an alternative family of models that we derived, in which we neglect the effect of facilitation between plants, and they will only compete. So the model equation now that we derive is much simple, because we are only looking into one species. We are only looking into the density of biomass, which is here rho, and we will only have two processes. The first one is death, so we assume vegetation dies at a constant rate alpha, and the second one is growth, which is going to have some other mechanisms. So basically, for the growth, we assume a logistic growth. So here we have, I have to say, sorry, that this is vegetation biomass normalized to 1. So basically, 1 is the maximum amount of biomass, normalized to the current capacity of the system. So we have some growth here given by beta rho, and then we have some limiting term that is basically going to introduce this limitation in the growth, given the finite space in the system. And the new term that we are going to introduce is a probability, here, PC, that is going to give us the probability of seeds to establish and give rise to a new plant. And that probability, the only requirement that we're going to impose is that it has to be a decreasing function of the amount of biomass that there is in a neighborhood around the position where the seed lands, basically. So here, rho tilde is going to be an spatial average of biomass, where G is the kernel function that is basically going to define the size of the neighborhood. So with this simple model, we can show analytically that patterns may form, and we can also calculate again the phase diagram that I was showing before. But now, even though we still recover the same sequence of patterns as we move across the amount of rainfall, across the intensity of the competition for resources, now the transition into a desert state is not discontinuous anymore, but now it is continuous. So what is this telling us? This is telling us two things that I think are quite important. The first one is that these patterns can be obtained via different mechanisms, and therefore it's worth to work on trying to figure out which ones are actually coming into play. But the second one is that patterns that seem different may have patterns that seem identical may have very, very different implications at the ecosystem level. So putting the two phase diagrams together, what we have is that we have the same sequence of patterns that is, the same sequence of patterns as we move across a gradient of rainfall. So these patterns actually, they have some potential importance to be used for ecosystem management. But in order to obtain that predictability, we need the models not only to recover the pattern, but also to do it from the right mechanisms. So recovering the pattern is not the whole story. We actually need to go into the underlying mechanisms and let those patterns to emerge naturally from these interactions. So even if our final question is trying to understand the consequences of these patterns for a given ecosystem, it's worth to step back first. And it's not actually worth, it's necessary to step back first and actually ask what are the causes of those patterns. What are the mechanisms driving the formation of those patterns? And this is the kind of approach that we started working on the examples that I'm going to show you now in a while. It's basically on developing mechanistic models of the individual level interactions that are driving the emergence of these self-organized structures. And then with those models, both the pattern and the implications of those patterns are going to emerge naturally. And it turns out that in order to start working on this approach, microbial systems provide us with a very, very good work in which we can try to test this approach, basically because they self-organize very quickly, so the time scales in which these patterns form in microbes are much shorter than in larger scale ecosystems, and also because we can manipulate the conditions in which these patterns form in the lab. Yeah.
Could you have been able to guess the qualitative pictures you are getting before running the simulations?
So here, the other way.
Opposite direction. Yeah, that's it.
For our model, we can actually do a linear stability analysis, so we can know in which region of what are the conditions to have patterns, and for for a particular set of PC functions, we can actually calculate the shape of the transition. That's one of the advantages of having these only one species models is that they are analytically much easier to work with. And something that we are trying to get now is go a little bit beyond the linear stability analysis and see whether these patterns are actually stable. So we are trying to do a weakly nonlinear stability analysis on the patterns to see if in both cases they are stable.
But I think the question is, how did you come up with this model in the first place? How did you know that such a simple model would give rise to patterns?
Well, there is studies on non-local competition models where they show that particles that only interact through non-local competition may give rise to patterns. So that was first intuition. And there are also another family of models that actually don't recover the whole picture, but they also use these kernel functions to study pattern formation in vegetation systems.
Quickly just go back and show us the equation that you...
This one? Sure.
So actually,
Yeah, sure.
Oh, now that I see it. So this first term is a non-local dispersal, but then it's modulated by a logistic equation, which I usually think of as a growth equation. So I just want to understand mechanistically, are you joining together dispersal and growth, or are those separate processes? Whereas, because even if I have nothing dispersing to me, shouldn't I have local growth?
Yeah, that's a great question. So actually, the dispersal is local, so it's modulated by this 1 minus rho. And this probability, what it's telling us is basically, so if we have a picture where we have some vegetation here, it's basically once you have a seed here, what's the probability of actually giving rise to a mature plant? And the reason for having the non-locality is because here, although you don't have vegetation, the extension of the roots is what matters, right? Because the competition is mediated by a long distance.
So the dispersal is local, but the competition is non-local. That's why you have this.
Yes. And I don't know if that answers your full question, because... Oh, perfect. Thank you. So, as I was saying, microbes provide us with this very good framework to test this approach. And basically what I'm going to show you now is two examples in which we can try to formulate these mechanistic models for the evolution of multicellular structures in two different contexts. So the way, I mean, there are two general ways of evolving these multicellular structures. The first one is when cells fail to divide perfectly from each other, so it's a kind of cells stay together to each other. And the second way is via an active aggregation of entities, so cells somehow come together with each other. So for the first case, for the staying together scenario, bacterial biofilms are a very good system in which this is happening. And very briefly explain the reason why this is a very good scenario for this staying together multicellularity. It's by, I mean, we can understand that by looking into their life cycle, basically. So the life cycle of biofilms starts with some planktonic cells that are free swimming in the external media. And once they find a surface, they attach to that surface. And once they attach to that surface, they start growing on the surface in a two-dimensional, forming a two-dimensional pattern. But as they grow in the surface, then the forces between cells are going to drive the formation of a three-dimensional microcolony in which the density of cells is going to increase, and cells are going to start secreting different molecules that basically are going to constitute an external matrix that is going to hold all the cells together with each other. And as this process continues to happen, then the microcolony grows in the third dimensional as well and gives rise to what is called the mature biofilm. So in this mature biofilm phase, the density of cells is so high that cells start coordinating their behaviors with each other. They start secreting molecules, and they start behaving more as a kind of community rather than as independent entities. And then eventually the mature biofilm is going to break down, and cells are going to be dispersed, and eventually they will find a new surface, and this life cycle continues. So because cells secrete this matrix, and they don't move when they are embedded in the matrix, then the spatial structure of this biofilm is very important in order to determine the functionality of this community. And this is something that is well known. But despite the importance that this spatial structure has for the functioning of the biofilm, there is much less work that has tried to understand how this spatial structure emerged. And this is actually what I'm going to show you today. I'm going to show you some work combining experiments and models in which we try to understand what are the interactions between the environment and cells that is going to drive the spatial structure of emerging biofilms. So basically, going back to the question that I was introducing in the beginning, first, we need to understand what are the mechanisms that are causing this biofilm spatial structure, and then we can move forward and try to understand what are the consequences of such a spatial structure. So very briefly, so you get a sense of the kind of experimental data that we had. We actually, Kari Nadel did it. We did a series of experiments in which we basically produced different strains of a biofilm-forming bacterium. In this case, we work with Vibrio cholera. And we produced these strains in a way that we have cells that are going to have high adhesiveness, cells that are going to have low adhesiveness, and for each of these, we have different colors. So at the end of the day, we have 4 different strains with different levels of adhesiveness and expressing different fluorescent proteins. So for our experiments, because we are interested only in looking into how the external media interacts with a given bacterial trait, we are going to mix cells that have the same adhesiveness and are only different in their colors. And once we created those mixes, we introduce them in a microfluidic chamber, we feed them from above, and we allow them to grow in that surface and colonize it, basically. So at the end of the experiment, we have these kind of 2-dimensional patterns in which we basically have either clusters of either red and blue cells with the same level of adhesiveness, the same degree of adhesiveness. And we can do those experiments by varying basically the 2 traits of the bacteria that are going to govern these surface occupation processes. And those traits are, as I was saying before, adhesiveness, and the second trait is the initial density of cells that is going to give us a proxy of how much these biofilms invest in dispersal. So when we did these experiments for low adhesive cells, we find that the patterns, at least qualitatively, I'm going to quantify them in a while, but just so you have a sense, qualitatively, they don't look to depend on the initial density of cells. But when we move forward to high densities of cells, we observe that the clusters of cells, the sizes of the cluster, depend on the initial density of cells. So we have this experimental result, and in order to understand it, we are going to develop a very simple ecological model trying to account for the minimum set of mechanisms that is driving this surface colonization process and see how successful we are in obtaining those interactions. Yes?
Just for context, are these all taken at the same time after initialization?
So they are taken when the whole surface is colonized, but the adhesive cells, because they divide more slowly due to the production of the matrix, they require more time to colonize the surface. We stop when the 2D surface is colonized just to avoid the 3rd-dimensional to start mattering, basically. Yes.
In the middle and then it goes out, or is it, how does it start? It starts just in one point on the surface?
So we basically have the microfluidic chamber that is like a rectangular thing, and the bacteria are provided from an external flow, and basically they randomly fall in the chamber, and they grow. So actually, I don't know if you can see from the movie the initial. Well, this is how, I don't know if it can be seen from there, how the red cells look like in the beginning. The blue ones, they are not shown here probably because of the contrast, but it's like a random initial distribution of cells. Yes?
I missed something very serious here. How does this experiment connect with your initial explanation on the formation of patterns of bushes in the Namibia desert?
Yeah. So the whole point is the patterns in the Namibia desert were a motivation to highlight the importance of actually trying to get the mechanisms. And at the end of this example, I'm going to highlight again the importance of actually getting the mechanisms in this system, and kind of trying to discuss how we shouldn't stop by reproducing the pattern, but trying to be more mechanistic and reproducing the actual interactions as well. So it's kind of, that's why I put in the title that vegetation was kind of a motivating example, because it's what drove the way of thinking, basically. So as I was saying, to try to understand that experimental result, we try to build this minimalistic model of surface colonization that is going to have only actually 3 ingredients. The first one is cell reproduction. So basically cells divide with a given rate. The second one is local competition for space. So basically once cells that are in crowded environments happen to divide, they are going to push each other. And the third force that we're going to have is cell relocation induced by external flows. So basically, the model that we are constructing here is an individual-based model. We have our surface is going to be a matrix. Each of the places in the matrix is occupied by a cell of different colors, and we are going to update the positions of dot matrix with these probabilistic rules. So again, the patterns that I'm going to show you, I'm going to discuss them qualitatively. I'm going to be quantitative in a minute. Yes.
Question on the rules. So from these rules, these are all local rules, except one could be non-local, the non-adhesive pushing. How many cells can it push? It can only push one cell, or can it push an arbitrary number until there's an open space?
So we try to model in principle, the cascade of pushing events that it should happen. It doesn't seem to be very important, and we stopped only with one cell. So basically, when there is two cells, we assume that they jump and they are removed by the external flow. We did a sensitivity analysis on that, and it doesn't seem to matter a lot. So again, if we do these experiments, assuming that cells are non-adhesive, we obtain that, our result is that the shape of the patterns doesn't seem to depend on the initial sell-off densities. But if we now change the adhesiveness of the cells, then we obtain that the shapes of the patterns now again correlate with the initial density of cells. But as I was saying, this is all qualitative. It's going to give us a kind of a vision that we are capturing the right amount of interactions, but we need a measure to actually see how good our model is. And the metric that we are going to use is the correlation length. So the correlation length that we obtain from the correlation function is going to tell us what is the probability of having two cells of the same color at a given distance in the matrix. So as you can see from these two examples, when the clusters of cells are larger, then the correlation distance decay more slowly because there is more probability of having cells of the same color at larger distances. But when the clusters are smaller, then the correlation function, represented by the black curve, decays very sharply. So with these correlation functions, we can define the correlation length as the distance at which this function crosses zero by the first time. And this is
exponential. It's not exponential. It doesn't look very exponential.
So for when the clusters become larger and larger, you kind of get a worse fit into an exponential. When the clusters are smaller, it is an exponential decay. But this is something we did all numerically, so we don't have an actual expression for a correlation function. But when you plot it in a log linear scale, you kind of get what seems to be an exponential decay. But we have no theory so far for this. So once we have this correlation length, then what we can do is we can calculate it for all our experimental patterns and for all our simulated patterns and see whether the model and the experiments agree or not, basically. And when we do that, here every small point is an experimental correlation length, and big symbols represent the mean correlation length obtained for different replicates of the model. What we observe is that the agreement between the model and the experiments is not only qualitative, but it's also a very good quantitative agreement between both the experiments and the theory. So now that we have a mechanistic model that is explaining the formation of these patterns and their relationship with bacterial traits and external forces, then we can use that model to explore other conditions that with the experiments are not so easy to get. So basically, we can extend our analysis to more initial densities of cells and to different flows intensities. And then for this, moving in this two-dimensional parameter space, we can see whether this dependence between cluster size and initial density is kind of independent of the environment or whether it is not. So what I'm showing you here is the difference between the correlation length for a pattern made by adhesive cells and a pattern made by non-adhesive cells. So when that difference is positive, so when adhesive cells create larger clusters, this is larger than zero, and therefore we should be in the red region. And when non-adhesive cells produce larger clusters, this is negative, and this is represented by the blue region. So what I'm telling with this result is that actually this dependence of the cluster size with initial cell density is not it's not universal, if you want to think in that sense. So we shouldn't expect adhesive cells to universally produce larger clusters than non-adhesive cells. It is actually a complex interaction between the external flow and bacterial traits what is driving this surface colonization process. And the implications that this result may have for the functioning of the biofilm are actually quite important, because as I was saying in the beginning, within biofilms, bacteria communicate with each other via the secretion of molecules. So the identity of the neighbors around each bacteria is going to be very important for the evolutionary stability of those behaviors, basically. So in order to try to understand the evolutionary stability of those behaviors, we actually need to have a good model that explains us how this spatial structure forms. And this is something that we are trying to work on. We are basically trying to see how these surface occupation traits are co-evolving in different environments to lead to different spatial structures, and how that's driving the evolution of different secretion behaviors. And of course, for different behaviors, it might be important for these different strains to mix with each other. For instance, if we have cross-feeding, if we have mutualistic interactions, then we want the species to be mixed with each other so they can interact. But there may be other types of interactions, as for instance, toxin secretion, in which cells, it's much better to have them segregated, or even this kind of cooperation, public good production, in which, again, cooperative cells are going to be more successful if they are surrounded by more cooperative cells. So in principle, these important interactions between external media and bacterial traits are quite important in order to determine this spatial structure and also to study the functioning of these microbial communities. But as I was saying before, this discussion on biofilms is only one of the ways in which we can get this self-organized multicellularity in microbial communities. The other one is this kind of coming together that I'm going to discuss using Dictyostelium as a model organism. And the reason why Dictyostelium is a very good model organism to study this question is because, again, it has a life cycle that combines a unicellular and a multicellular phase. So Dictyostelium cells, they live as single cells. They are unicellular organisms. They feed on bacteria, and they reproduce by cell fission. But once they consume all the resources in the environment where they live, they start signaling with each other. They start synchronizing their behaviors, and they start actively aggregating to form a multicellular structure. So in this movie, you can see that process. The multicellular structure is forming here, and all cells are somehow migrating to that position. So once this multicellular structure forms, it's going to undergo a developmental program that is going at the end to culminate in a formation of a fruiting body. And because of that, Dictyostelium has been used as a model organism to study evolution of multicellularity, to study evolution of cooperation even in microbes. But I'm not going to go into that phase. I'm going to stop in this first phase here, and I'm going to quantify the interactions that are driving this aggregation process, and I'm going to discuss the degree of synchronization that these cells are showing in this phase. So as you can see in this snapshot, or even better here, although we have already a multicellular structure form, there are some cells that haven't joined this collective behavior. So there are some cells that are staying solitary. They are out of the multicellular phase, and those are the ones that I'm going to be interested in the study. This is the ones I'm going to discuss with you today. So these cells, we know from our previous work and also from some theory we developed, they don't die once they don't aggregate. I mean, if food comes back to the system quickly enough, they can reproduce again and they can recover the multicellular phase, so they are viable for some time. So in principle, they are good candidates to have something to be meaningful for the life cycle of this organism. Because you can imagine a scenario in which nutrients recover quickly enough, so therefore not being part of this collective behavior, it's actually beneficial because you avoid all the cost of involving in this developmental process, and you can start dividing much quickly than the cells that become part of the aggregate. But despite that potential, we do not know empirically if these cells are an actual trait of Dictyostelium. And this is the first experiment that I'm going to show you. So we in order to see if these cells are actually meaningful, the first thing that we need to ask is whether natural selection can act on them, basically. And in order to try to address this question, we did a series of experiments. Actually, Fernando did them. He's a grad student at Princeton, where we counted the number of cells that don't aggregate, so the number of these loner cells that I was highlighting before, as a function of the number of cells that were initially plated in the system. So when we look into this data, we observe that if the initial density of cells is very low, aggregation doesn't even start, and therefore all cells remain unicellular. So this is a one-to-one slope, nothing in principle very interesting here. But if we move beyond some threshold, then aggregation starts happening, and we observe that the number of cells that don't aggregate is independent on how many cells there were initially in the system. And if we repeat this experiment for different genetic variants of this organism, we observe that different strains show different levels of investment in non-aggregation versus aggregation. So basically, what we are showing with this experiment is that there is variability, and is that the density of loners is actually a variable trait among different strains, and it's also a heritable trait. But as we saw in the morning, in order to show that natural selection is acting on this trait, we also need to show that this variability and this heredity is leading to fitness differences. And this is something we haven't been able to show so far, to be honest. But still, we are getting something interesting for this experimental result. What we are observing is that there is, yes?
So can I just ask a naive question? So why are the points so spread? Why isn't it a smooth curve?
The experimental point?
Yeah, the red ones, for example. You have such large numbers, shouldn't...
So each of these points is an independent replicate of the experiment. So basically the way Fernando did these experiments is he puts cells in a...
It's just one experiment.
Each point is one experiment. So for each point, for each experiment, we put cells in a plate, we allow them to aggregate, and then we count the number of cells that don't aggregate. And that's 1 point. And then we repeat that experiment many, many times. And each of the replicates is 1 point.
Okay. I mean, because the red one doesn't look like... You need some imagination to know what the red one is, right?
So the red one...
to say those red dots are a constant? I don't know.
So the red one, the thing is that has more natural variability, so the process has more noise. The distribution is wider. But if we manipulate the conditions of the experiment, we can reduce that noise, in principle, a lot. But the problem is that if you reduce that noise, you also reduce the number of cells. Because basically, if you change the conditions in which this aggregation happens and you favor aggregation, then the process is less noisy, but also leaves less cells because they aggregate better they communicate better with each other basically, and they aggregate better. This is like the one slope. This has different. So these are experimental points, and this is the linear regression to show that this is all cells don't aggregate, basically, and whatever you put in the plate, they remain without aggregating. You need some quorum, basically, for these cells to trigger the aggregation. So, again, as I was saying, these results are revealing some complex interaction between the environment and the cells in order to drive this experimental result. But we don't really know from this experimental data what those mechanisms are, what those interactions are. So going back to the questions that I've been trying to ask for each of the examples that I was showing you today, first we need to address the mechanistic causes. We need to understand how this pattern form, this self-organized process form, and then we will be able, with that model, to see what are the consequences of that self-organized process. And in order to do that, we generated a random walk-based model in which basically we are going to represent each cell by a random walk that is going to either aggregate or not, depending on the conditions. I'm going to explain the model now. So our proposal is that these loners are the result of the aggregation process needing some quorum, because as I was showing you before, when the initial density of cells is very low, aggregation doesn't happen, so we know we need a critical mass of cells to start the aggregation. And then what we propose is that these loners emerge because the response to that quorum is stochastic. So what does it mean? First with figures and then with equations. With figures, what we propose is that initially, when all the cells are plated in the system, they all signal with each other, and because the initial density of cells is very high, they all are likely to start doing this aggregation process. But because this aggregation, I mean the starting of this aggregation is stochastic, there will be only some cells that will actually commit to the aggregation and will start moving towards the aggregation center. And as they start moving, they will create spatial heterogeneities in the density. They will create some regions in which the concentration of signal is going to be low, and therefore cells are going to be trapped there without the possibility of starting the aggregation process. And at the end, as this aggregation process, I mean, as cells continue to migrate and continue to migrate, we will reach a stationary state where we have some cells that aggregated and some cells that did not. And in our model, these cells are going to be the loner cells. So with this model, if we run numerical simulations of this model, of course, the way in which this population is going to partition between aggregated and non-aggregated cells is going to depend on the way that we parameterize the model. So we may obtain different levels of aggregated cells versus non-aggregated cells. But what is very interesting is that, at least qualitatively, we recapitulate the two phases in this curve for the loner density as a function of initial cells. So in the beginning, we have non-aggregation because we have that quorum that needs to be met for the collective behavior to start. And then we have this density independence behavior in the loners. But this is all like a very computational model. This is all simulations. And then the question that is really interesting is whether we can come up with an analytical explanation for the origin of these cells. And in order to try to obtain that analytical explanation, I want you to think in a case in which we assume that the diffusion of these molecules is somehow infinite. So basically, cells are able to communicate all over the space. It's a kind of a mean field approach in which we can map the concentration of cells to the concentration of signals, basically. So in that case, our model reduces to a simple model of three populations in which we have pre-aggregated cells that are going to undergo that transition to aggregating cells, and that transition is going to depend on the concentration of signals, as I was saying. So for this first transition rate, we need some quorum. This is basically a theta Heaviside function. And then for the second transition, to aggregated cells to multicellular, to the multicellular state, because we're assuming that cells are able to communicate all over the space, we can define an effective transition rate that is going basically to be the interplay between the velocity of the cells and the system size. So we have these two temporal scales that are defining the two transitions from pre-aggregation to finally the multicellular phase. So if we take the deterministic limit of this stochastic model, we obtain this set of two differential equations. Actually, we have a third one that can be removed because the total population of cells is conserved, in which we basically have transitions between the populations. I mean, this looks like a very simple model, but I want you to pay attention in the fact that this lambda here is not constant. It's actually a function of P and A. So it's not so easy to solve. We actually cannot solve analytically this for any condition. But what we can do is we can do some asymptotic analysis on this model. We can basically try to see what happens when the cell density is infinite and see whether we get some analytical result for those plateaus in the number of loners that I was showing you before. So doing that calculation that I'm not going to show all the steps, but what we get at the end is basically that the density of loners is the result of the interplay between these two temporal scales that are not synchronized in a perfect way. So basically, when the transition from the first state to the other is much slower, when the transition from pre-aggregation to aggregation is much slower than from aggregation to multicellularity, then the number of loners is not equal to 0. But then, as we have in the other regime, the number of loners basically vanishes, which can be summarized in this figure. Here, the line is the analytical calculation, so it's basically this phase transition, And the dots are numerical simulations of the system of ODEs, in which we basically, as we increase the number of cells, we collapse to the analytical solution. And finally, just to finish very briefly, as I've been discussing, going from these behavioral scale processes to the ecological and evolutionary scales, what is this behavior telling us? So these loners are actually something that could be very important for the ecology of this organism. And the main reason for that to happen is because slime modes are very, very diverse. So even on very small scales, we have many different strains coexisting with each other. And as we also saw this morning, when different strains start together, they don't discriminate with each other, and they can form a multicellular structure that is made by cells with different genetic origins. And when that happens, what we can measure both in the experiments and with our model is that the behavior of the strains, this is just a hand-draw plot to show you that when this happens, the number of loners of these strains changes. So basically, due to these interactions, cells are shifting their behaviors. Red cells don't behave in the same way when they are surrounded by red cells or when they are surrounded by blue cells, saying in other words. And in order to test the implications that these shifts in the behavior may have for the ecology of the organism, we can actually use our model and run it through many different growth and starvation periods. So go to an ecological scale with our model and see how the patterns of diversity are affected by these interactions. So very briefly, what we find is that if we don't have these interactions and we run our model on different patches, different realizations, it doesn't matter the proportion of the cells. At the end, the proportion of cells belonging to each strain, at the end, all the patches are going to be dominated by one single strain, and therefore the diversity of the species is very low. But when we allow for these interactions to happen, then we observe that the diversity of the species increases a lot due to these shifts in the behavior, basically. So, summarizing very briefly this project, and also trying to go back to the bigger picture that I was introducing in the beginning with these vegetation patterns, the way that I'm looking into these self-organized processes is by understanding them as a link between between processes and interactions that happen at the individual level, between behavioral scale processes and larger scale processes that happen at the ecological and evolutionary scale. And of course, the way these self-organized patterns form depends on different interactions, both between individuals or between individuals and the environment, and they may affect reproduction, movement, they may affect reproduction, but as a whole, they all provide this link in order to understand the transition between behavioral scales and ecological scales, and therefore I think they are a very powerful tool to try to understand how biological complexity may be evolving. And with this, I want to finish thanking all my collaborators in each of the projects. This is the Vegetation project, the biofilms, and the Dictiostellium, and also the funding. And thank you for your attention.
So I have two questions. So at some point, you want to make a prediction. So for example, for these slime models, suppose you start with mixed density where it's not constant. Some regions have low density, some regions have high density. Can you make a prediction of what's going to happen?
You mean if in the system I have regions with different densities? Yeah. In principle, it's going to depend a lot on how, well, provided that regions are able to communicate with each other.
Sure.
No, I mean, then what I would expect is the density of loners to be also heterogeneous in space, because basically regions with higher density will aggregate and they will leave their loners. No, actually, in principle, provided that the initial density of cells is always above the threshold, you should expect the density of loners to be always the same. Because at the end of the aggregation process, the number of cells that you count doesn't depend on the cells that you had in the beginning.
Okay, I guess what I'm asking is I would like to put more parameters into my experiment, so because your model has some parameters in it, right? Mm-hmm. And to make a prediction, I need to have my my experiment has to involve more parameters than there are in the model.
So the best prediction that you can make, and actually that was the one we make, is when you go into the interactions between strains, we basically, in the model, we assume that different strains signal in different ways. So basically they release more molecules. And when you allow that aggregation to happen in mixes and you count the number of loners, you obtain some, so basically, if I count the loners as a function of the percentage in the mix of each of the strains, our model predicts this to be a kind of a sigmoid curve.
Okay.
And this is what we obtain in the experiments.
Okay. So the other question was, you had at some point you had some quantitative experiments. So it was early, maybe 1/3 through your, it was before you discussed the slime model. You had some quantitative analysis and you said your model matched the quantitative. Yeah, that one. So, sorry. I mean, can you explain, like, for why at 10 to the -1 you have all these experiments all over the place and your
Yes.
model has...
So, do you mean why we have more data for here than here? Or?
No, it just doesn't look like your model fits very well to the experiment. Your model predicts 1 point at 5 when I'm at 10 to the -1.
The model predicts
Yeah.
5 something.
Yeah, but your experiment has things all over the place. So how can you say that?
Yeah, so this is because the large symbol is the mean correlation length over different model realizations, and every small point is an independent replicate of the experiment. But actually, so this is when we reconstruct the distributions, both for the experiments and the model. And actually, the one that seems in that figure to be the worst is the better, because basically we have more experimental data. So here, basically, the dashed lines are the reconstructed distributions for experimental data, and the full lines are for the model. And here it seems, well, the blue one it's a bit worse, but the green is the most satisfying. Thank you.
Yeah, I had a follow-up. If you could go back to the same slide on the correlation length, just
Yes.
to flip back where you were. I realize that the way you've done this, oh, it's okay. The way you've done this experiment is you keep saying that you stopped the experiment when it got full because you didn't want to go to 3D. On the other hand, when I see these, I think of coarsening dynamics, and I'm just wondering if you've either in the model or in your experiments not stopped it and asked the question, at what rate the correlation length evolves with time, and is it indicative of something like coarsening or other spinoidal decomposition or other kind of features that we might expect?
You mean as the clusters grow in 2D, or once we?
No, even in 2D in your model, you could restrict it. You don't have to have a 3D component, or if you're able to increase flow rate so the cells out of the plane disappear.
This is something that actually, I think I don't know if you collaborate with David Juncker, he pointed it out from Georgia Tech in a conference, and we are trying to get the field equation for this lattice model to see whether we can get critical exponents for the surface growth, and actually see whether there is some theory for that.
They are my colleagues, but I don't work with them on that project. But then a follow-up question: the Vibrio can do lots of things to each other. So these blue or red strains are reporters; they're equivalent. You think they're equivalent? Right?
Well, actually, Kari did a previous paper with this, and he showed that the fitness doesn't change for.
Got it. And so my quick question is, do they stab each other using these type 6 secretion systems? I'm sorry to go into the molecular details, but sometimes they can antagonistically kill each other locally. Is that happening here? Or you
No,
think
it's not. Here it's not happening.
You're sure these don't have a secretion system?
In the experiments that we are doing, they don't.
I have a simple question. So in this plot, the model has arrow bars and the data does not have arrow bars. I don't understand why.
So the data doesn't have error bars because it's not any average. It's only every point is an experimental data.
Yes.
So the experimental error is so basically the error bars here are statistical. It's not an error bar. It's the variance of the distribution that we are recovering from the many simulations.
Yes.
And the experimental data is only 1 point, so we don't have.
So you're just doing many simulations, and the error bar comes from the variance of
the variance of the final list. It's basically, if we go back to the.
But let me ask you this: if you do, if you do infinite number of simulations,
yes,
the error bars go to 0 or not?
These ones don't because it's I have a distribution of correlation length.
Distribution of correlation length.
Which is basically going to be given by the.
But it's not given by the model. The model doesn't give you the.
So what gives me the variance in initial distribution is basically comes from the different initial configurations that I may have because I'm starting them randomly, and at the end I have some variability in the distribution.
Okay. So is the model stochastic or deterministic?
Yeah, no, it's a stochastic. Sorry, they reproduce with some given rate, and cells are relocated with some probability as well.
And in the equations, I didn't see any stochasticity. Where are they? Can you show me the stochasticity?
Well, yeah, sorry, because for these ones I didn't put the equations. So basically, the stochasticity comes from the rates at which each of these processes happen. So the structure of this model is a lattice model, like kind of an IC model.
So in the equations, there were rates. So the rates were stochastic, that's what you're saying? Or?
Yeah, so basically each process happens with a given rate. So the probability for each process to happen in a time step is the rate times the length of the time step. And then I basically calculate the probabilities for each update to happen, and then I sort a random number, and then depending on the probabilities, I update either one process or the other. So the dynamics is stochastic. It's more kind of an updating rule for an IC model or for, I don't know, a contact process model that people use for epidemic spreading, or it's more that kind of architecture.
Yeah, so can you put a slide where you show aggregation based on stochastic quorum sampling, please?
Yes. You mean the model or?
30. Yes. So maybe I'm just wondering, I would expect that even if it's stochastic, the probability of successful quorum sampling would depend somehow on the distance from the origin of the signal. Mm-hmm. You know, is that what is happening, and is that included in your model? Because it seems like you're decoupling, like accepting the signal from the migration, which I think it's related.
So in the full model, we don't do that. When we take this kind of mean field approach, in which we assume that every cell interacts with every cell with the same intensity, we are doing that just to try to get an analytical result. In this case, we are not. We actually have, we assume that each cell is signaling at a constant rate, and that signal decays, and that gives you like a profile with the distance, as the one that you have in mind. But what we have as a binary variable is basically the response to that signal. So if at the position of a cell, the concentration is above my response threshold, then cells start aggregating, and if not, they don't. But you are incorporating the connection with the distance through the decaying profile of the signal, basically. So if you are if you are a cell and I am a cell, and you are emitting from there, and you are emitting there and I'm sensing here, I will sense more signal than if I move here. And we are incorporating that, at least for when we want to simulate the whole process. When we do the analytical limit, then of course we relax that, because otherwise we cannot get the results. And just to finish, the way the cells distribute in the system is like this. So cells that don't aggregate always are farther from the multicellular structure because of that, because they don't sense, they stop sensing signals earlier.
So you go stationary, or when do you stop?
Mm-hmm.
Time will review.
become stationary, or you stop sometimes?
Yeah, no, this configuration is stationary basically because at some point cells in the multicellular stage don't aggregate, and that's something we know. So at some point, the concentration of cells moves below the threshold for aggregation, and then the configuration of cells is frozen. And that happens in the experiments. Of course, the mechanism to stop the aggregation process is way more complicated than this, but the configuration is also like stationary. At some point, the multicellular structure starts doing their development, and the non-aggregated cells remain in the...
Also for the correlation lengths, you also stop when you measure correlation lengths.
So for the correlation length, we need to stop by hand because otherwise they start growing in 3 dimensions, and then the experiment messes up. Actually, that's another project that one of the collaborators was doing, was looking into competition in the 3 dimensions, and he sees that one displaces the other depending on different traits, so that would be like a different story, I guess.
When you see some plateau or something leveling off, or where it's reasonable to stop, or just because you can't?
We stop when basically when the pattern grows in the whole surface, we stop. We assume that that's like when the 3rd dimension is going to start mattering, and then we stop we take the picture and we stop the experiment.
Yeah. Can you explain again why you cannot calculate averages and variances for the data in that plot?
In this one? Sorry. In this one?
No, the one we were discussing before with the simulations against data, yeah. That one, yeah.
Here? No, here you can, in principle, yeah. Yeah, actually the one this figure that I show you after, it's the actual reconstruction of the distribution for those same data.
Okay.
So the dashed line is the reconstruction of the distribution for the experimental points, and the continuous line is for the model simulations.
Okay, so thank you again. And,
No, I don't have anything to say. So just for people who didn't get the email, tomorrow the seminar will be by Danilo Liarchi, and Daniel will be on Thursday.